Okay, let's continue the surface chemistry. Differentiate between the homogeneous and heterogeneous catalysis. Homogeneous catalysis means when a catalyst and the reactants are in the same phase. Catalyst and the reactants, if they are in a same phase, such reactions are called uh, homogeneous catalysis. Suppose uh, reactant and the catalyst are in different phase, such reactions are called heterogeneous reactions. One example for the homogeneous catalysis, nitric oxide gas catalyzes the combination of the most of the sulfur dioxide and oxygen in the lead chamber process, and this process is called a contract process. And here, lead will act as a catalyst and converts sulfur dioxide, water, and oxygen into sulfuric acid. Here, the catalyst is nitric oxide. Nitric oxide is a nitric oxide, oxygen, water, and sulfuric acid are in same phase. This gaseous one is converted into a liquid as the product. So the catalytic process in which the reactants and the catalyst are in different phases is known as the heterogeneous catalysis. Oxidation of sulfur dioxide into sulfur trioxide is an example for heterogeneous catalysis. And here platinum is the solid, sulfur dioxide is a gas. And these two are in different phase. Obtained the product is sulfur trioxide if it is a gas. Let's look into the uh, what is enzyme catalysis? Give example. Enzymes are biological catalysts. Enzymes are also proteins that are folded into complex shapes that allow smaller molecule to fit into them. The place where these substrate molecules fit is called the active sites. Uh, one example of this one is the inversion of sugar by invertase. And you can observe that sucrose is a disaccharide on hydrolysis in presence of invertase enzyme converts a sucrose into glucose and fructose. So conversion of the glucose to ethyl alcohol and carbon dioxide. That means degradation of the glucose in presence of zymase. Zymase is an enzyme. It converts a glucose into ethyl alcohol and carbon dioxide. These two are the examples of the enzyme catalytic reactions where there is a conversion of sucrose into glucose and fructose by the invertase, and zymase which converts glucose into ethyl alcohol and carbon dioxide. Let's take another one, the autocatalysis. So in autocatalysis reaction, no specific catalyst is added. Instead, one of the products acts as a catalyst and increase the rate of the formation of the product. Decomposition of arsine. Arsine means ASH3 is called arsine. Is formed by the arsenic formed in the reactor is autocatalyst. So that means arsine formed arsine will act as a autocatalyst in this reaction, which means that once the arsine is formed, then the reaction proceeds faster compared to the previous one. In this process, arsenic AS act as a catalyst. One more example, potassium permanganate on reaction with sulfuric acid and oxalic acid. Here there is a formation of manganese sulfide. Once manganese Mn2 plus is formed, this will act as a catalyst and reaction completes. I will go to the next slide. In the next slide, I'm taking explain how the intermediate compound formation theory can be used to explain the catalysis. This theory explains the mechanism of homogeneous catalysis. According to this theory, the catalyst reacts with one of the reactant to form an intermediate compound. The highly reactive intermediate compound then decomposes or combines with the other reactant to form a product. The catalyst is regenerated. So you can observe that A plus B giving rise to product is the reaction. And here a catalyst is not used, but in this A plus C, C indicates the catalyst. C catalyst react with the A forming an intermediate compound called AC. This AC react with the another reactant forming a product with the liberation of catalyst. In the second equation 2 and 3, a catalyst inward reaction, and here activation energy is less, and in the first reaction without the catalyst, activation energy is more. So that means catalyst decreases the activation energy, thereby it increases the rate of a reaction So with this, I will take right. Okay. 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 Right. Right. So here I'm showing the diagram. So this is the activation energy without the catalyst, and this is the activation energy with the catalyst. That means with catalyst, the energy barrier is decreases. Uh, that's the reason the reaction proceeds faster. And here I'm taking the examples of homogeneous reaction where the intermediate uh, ion theory. Sulfur dioxide react with oxygen, giving rise to sulfur dioxide. This reaction takes place in the presence of nitric oxide, NO as the catalyst. Um, Oxygen react with NO, giving rise to nitrogen dioxide. Reactant and then catalyst form an intermediate compound. This intermediate compound react with the another reactant, forming a product with the liberation of nitric oxide as a catalyst. So then I will take up, explain the theory of the acid-base catalysis. Uh, Acid-base catalysis means homogeneous catalytic reactions are catalyzed by acids, bases, or both. Such reactions are known as acid-base catalyzed reactions, and the phenomenon is called acid-base catalysis, acid-base reactions catalysis. For example, hydrolysis of ethyl acetate into an acid and an alcohol is carried out under the catalytic influence of acid. Ethyl acetate and reaction with water in presence of H plus gives rise to acetic acid and alcohol. So here, the mechanism of the acid catalyzed lies in the formation of an intermediate complex between the proton H plus and the reactant. This complex then reacts with the water to form a product, and proton is released. H plus and ester react with the H plus and ester, gives rise to an intermediate complex, and this intermediate complex breaks here, forming a two intermediate. One is a neutral product, another is unstable species. This unstable species further react with water, giving rise to acetic acid. Then I will take up heterogeneous catalysis. Heterogeneous catalysis is the best explained by the modern adsorption theory, which is the combination of the intermediate compound and the formation theory and the old adsorption theory. The following mechanism, the first step is the diffusion of the reactant molecule to the surface of the catalyst, and the second step is the adsorption of the reactant molecules on the surface. When the reactant molecules strike the catalyst surface, they are held up there by the force of attraction. Formation of the activated complex on the surface. The adjacent reactant molecules that are adsorbed on the surface from an unstable intermediate complex. Step 4, decomposition of the activated complex to form products. The unstable activated complex ultimately breaks to form a product. Decomposition of the product molecules from the surface. The product molecules diffuse away from the surface, making the surface of the catalyst available for fresh molecule to adsorb. This is about the adsorption theory mechanism. Okay. Then I will explain the Michaelis-Menten mechanism. Michaelis-Menten mechanism is a fundamental concept in enzymology that describes the kinetics of the enzyme catalyzed reactions. It provides the insight into how enzymes interact with the substrate to form a product. The mechanism is named after the Leonard Michaelis and Paul Menten, who proposed it in 1930. So the Michaelis-Menten mechanism, enzyme substrate complex formation. First step is the enzyme substrate complex formation, that is ES complex. The enzyme E and the substrate S bind together to form an enzyme substrate complex ES. This step is typically reversible. E plus S gives rise to ES. The product formation. Once the enzyme and substrate are bound in the ES complex, a chemical reaction takes place, resulting in a formation of the product P. This step is irreversible. ES gives rise to E plus P. Enzyme regeneration. After the product is formed, the enzyme is released and can bind to another substrate molecule to repeat the catalytic cycle. Then the rate of reaction. The rate of reaction can be described by the Michaelis-Menten equation, which relates the reaction rate V to the concentration of the substrate S and the other kinetic parameter. That is V naught is equal to V max into product of substrate upon Km plus concentration of the substrate, where Vmax represents the maximum reaction rate when the enzyme is saturated with the substrate. Km, Michaelis constant, represent the substrate concentration at which the reaction rate is half of the Vmax. It is a measure of the affinity between the enzyme and the substrate. Lower Km values indicates higher affinity. So then the Michaelis-Menten mechanism assumes that a rate determining step of the reaction is the conversion of the ES complex into product, and that the concentration of the enzyme remains constant throughout the reaction. The mechanism provides the useful framework for the understanding the enzyme kinetics and predicting the behavior of the enzyme catalyzed reaction. It helps to elucidate the factors such as the enzyme substrate binding, the formation of the breakdown of the ES complex, and the influence of the substrate concentration on the reaction rate. In the next slide, I'm taking when a graph of substrate concentration against the rate of reaction is plotted, the rate of reaction initially increases rapidly in a linear fashion as the substrate concentration increases. The rate then plateaus and increasing the substrate concentration has no effect on the reaction velocity. As all enzyme arrive, active sites are already saturated with the substrate. This is the graph. That means initially increases, later on there is no effect of increasing on the rate of a reaction. This is because of the this is because of the occupation of the molecule on the active sites. Then explain the important characteristics of the enzyme catalysis. Enzyme catalysis is a process by which the enzyme facilitate and accelerate chemical reactions in living organisms. Enzymes are biological catalysts that increase the rate of reaction by lowering the activation energy required for the conversion of the reactant into products. Specificity. Enzymes exhibit remarkable specificity towards their substrate. Each enzyme typically catalyzes a particular reaction or a specific class of reactions. This specificity arises from the precise three-dimensional shape of the enzyme's active site, which complements the shape and chemical properties of the substrate. The specific interaction between the enzyme and the substrate are crucial for the facilitating the reaction. Active site. Enzymes possess an active site, which is a region or a cleft on the enzyme molecule where the substrate binds. The active site has a unique structure that allows for a specific recognition and binding of the substrate. The binding of the substrate of the active sites forms an enzyme substrate complex and facilitate the chemical transformation of the substrate into product. Lowering the activation energy. Enzymes accelerate the reactions by reducing the activation energy required for the conversion of the reactant into products. They achieve the lower energy barrier by binding to the substrate. Enzymes can stabilize the transition states or facilitate the specific chemical reactions, allowing them to occur more rapidly and readily. Induced fit. Enzymes often undergo conformational changes upon substrate binding. The phenomenon as induced fit involves the enzyme adjusting its shape from more precise and optional binding arrangements with the substrate. The induced fit enhances the enzyme catalytic activity and helps in achieving mechanism. Enzyme substrate complex stability. The stability and the duration of the enzyme substrate complex are crucial for the efficient catalysis. The active sites binding interactions with the substrate, including hydrogen bonding, electrostatic interactions, and hydrophobic interactions, contribute to the stability of the complex. Optional binding, optimal binding ensures that the substrate remains bound long enough for the reaction to occur. Enzyme regulations. Enzymes activity is often regulated in response to the needs of the organism. Various mechanisms such as the allosteric regulations, covalent modification, and feedback inhibitions control the activity of the enzyme. Regulation ensures that enzyme catalysis is finely tuned to the metabolic demands and the environmental conditions of the organisms. Enzyme recycling. Enzymes are not consumed in the reaction, they catalyzed. After the reaction is complete, enzymes are released from the product, allowing them to reused for subsequent reactions. This property enables enzymes to function as an efficient catalysis, significantly increasing the reaction rate without depleting. Let us take what are unimolecular surface reactions? Explain. Unimolecular surface reactions, also known as the dissociative adsorption or Langmuir Hinshelwood reactions, refer to a specific type of chemical reactions that takes place at a surface of the solid surface. These reactions involve the dissociation of the single adsorbed molecule into multiple fragments on the catalyst surface. In a unimolecular surface reaction, the reactant molecules, often a gas, approaches the surface of the catalyst and absorbs onto it, forming an adsorbed intermediate. The adsorption process typically involves the weak chemical bonds, such as the physisorphism or chemisorption between the reactant molecule and the catalyst surface. Unimolecular surface reactions play a significant role in various catalytic processes, such as the heterogeneous catalysis. They are commonly encountered in reactions involving the hydrocarbon conversion, such as cracking, reforming, and hydrogenation, as well as the surface reactions involved in a catalytic reactions for air and water purification. It's important to note that unimolecular surface reactions differ from bimolecular surface reactions, which involve the interaction between the two adsorbed species on the catalyst surface. Unimolecular reactions specifically focus on the dissociation of the single adsorbed molecule, while bimolecular reactions involve the collision and interaction of the two adsorbed species, leading to a chemical transformation. In summary, unimolecular surface reactions occur when a reactant molecule absorbs onto a catalyst surface, dissociates into a multiple fragments, and forms a new products. These reactions are important in heterogeneous catalysis and have an application in various industrial segments. With this, I will go to the next slide. The next slide, once adsorbed, the reactant molecule undergoes a chemical transformation, often by breaking the chemical bonds within the molecule. This results in the formation of multiple fragments on the catalyst surface. This fragmentation can then interact further with the other adsorbed species or participate in a subsequent reaction steps. The rate of unimolecular surface reaction is influenced by various factors, including a concentration of the reactant molecules, the temperature, and the properties of the catalyst surface, such as its composition and structure. The activation energy required for the dissociation of the adsorbed reactant molecule is a key determinant in the reaction rate. Then explain the bimolecular surface reaction. A bimolecular surface reaction, it is assumed that two reactant molecule A and B are adsorbed on a neighboring sites on the catalytic surface. The order of reaction depends on the extent of adsorption by the two reactants. Both the reactants have low adsorption. In such case, both of the reactants are weakly adsorbed, results in the reaction being first order with respect to each of the reactant. Overall reaction is therefore second order. For example, the reaction between the nitric oxide and oxygen on a glass surface. Once the reactants has very low adsorption or as a strongly adsorption, one of the reactant is strongly adsorbed means that the reaction is first order with respect to the weakly adsorbed reactant. That means if A is strongly adsorbed, the reaction is first order with respect to B. There are two extreme possibilities now. At low concentration of A, the order is 1 with respect to A. At high concentration, the order is -1 with respect to A. The higher concentration of A, the slower the reaction goes. In this case, we say that A inhibits the reaction. For example, reaction between the carbon monoxide and hydrogen on platinum surface. Okay. Treatment industrial effluent. The discharge waste of the inorganic and organic compounds from the paints, dyes, drugs, paper, sugar mills are called industrial effluents. Some of these substances are highly toxic, such as lignin. Heavy metals like mercury, lead, cadmium, etc., which are non-biodegradable, they get accumulated in the body, living organisms. Though it is not possible to follow a general method for the treatment of industrial effluent, we become of the complexity and variety of pollutants involved. The primary and secondary treatments are used for the sewage treatment is to followed by the industries. Then the effluents from the wood industry contain lignin, cellulose fibers, and the grease, oils, and alkalis, heavy and other metal organic compounds are called as the black liquor. At least 50% of the wood is discharged into a waste. The color of the effluent is removed by treatment with lime. The black liquor is neutralized with acid. When lignin gets separated, the insoluble material is removed by filtration. Then the remaining chemicals are removed by activated sludge process by the oxidation with chlorine, by ultrafiltration or reverse osmosis. Activated sludge process is preferred by the treatment with the bleaching powder. In ultrafiltration, the solution is passed under pressure through a membrane containing pore size 2 to 10,000 nanometer. The larger molecules are retained by the membrane. In reverse osmosis, a semipermeable membrane of pore size 0.04 to 600 nanometer is used. In sugar industries, the effluents contain the waste of high biochemical oxygen demand, including sugar and proteins. This is the subjected to oxidation where the microorganism oxidized them into alcohol, carbon dioxide, and methane. So this is about the surface uh, chemistry.